Hello, everybody. We are the Carroll High School iGEM team, and our project this year is engineering E. coli to detect and destroy biofilms. So just a little introduction about the team. We are a team from Carroll High School, like I previously stated, of 2 graduated seniors and 8 seniors this year. So why we decided to take on biodiesel? The problem with biodiesel now is there's a high concentration of fatty acids and methyl esters compared to regular diesel. This acts as a food source for many different types of microbiology. The water condenses within the tank, and this creates a habitat for the microbiology, creating this fuel or this water layer that I'll talk about later. So as I was saying, with the microbiology, the problems that are created, and these consist of the fouling of fuel pipes. As you can see, we have a large pipe that is completely covered in gunk, and that is made up of different types of bacteria and fungus. We also have biocorrosion. These are 2 metal coupons that were corroded because of the fungus and bacteria breaking down parts of that material. And fuel degradation, which just hurts the hurts the combustion rate of the fuel. As we were just discussing this project with many of the other important people in this field, we talked to Dr. Nike at the Air Force Research Laboratories about some of the work he had done. They gave us a lot of different isolates to work with of different fungus and bacteria. This allows us to know what we're working with in these fuel tanks. We also went to the combustion burners manufacturer called Beckett. Beckett gave us a lot of things to worry about with why biofuels are such a problem in this day and age because of all those extra fungal the extra oxygen in the fuel creating a perfect environment for microbiology. We went to Marathon and discussed many of the things about why it is not the manufacturer's fault that a lot of these fuels are getting contaminated, but it is about the transportation and storage of these fuels. And finally, we talked to Scott Fenwick about from the National Biodiesel Board about why biodiesel is on the up and coming, and why he had many concerns about adding more microbiology into fuel tanks. He thought that adding more microbiology would just add another isolate that would eventually disrupt the tank even further instead of helping it. So as I was saying before, the water layer that condenses on the top of the tank and then moves down into the fuel.
Please report down to Mr. Taylor's room. Brady O'Blunnett, please report down to Mr. Taylor's room.
The problem is at this fuel interface that you see here. We are getting, in between the water and fuel, a mix of where the bacteria and fungus can feed off of the fuel, but still live within the water. So this was an SEM picture of that metal coupon that I was talking before. And as you can see, these long strains here are the fungus, and all the other stuff is the bacteria. So as you can see, we had a mix of fungus and bacteria, and this is essential into the design of our project. So Yasmin will talk about how we solved this problem.
So before I talk about exactly how we solved this problem, I want to let everyone know that we broke our project into 3 parts: a detect, a deliver, and a destroy part. So I'm going to talk to you more about the detect mechanism. So for our detect mechanism, we focused on using quorum sensing, which is the form of communication between microbes. We particularly focused on the microbe Pseudomonas aeruginosa, which is a common component found in biofilms. And that Pseudomonas aeruginosa excretes the quorum sensing molecule called N-butyl homoserine lactone, otherwise known as C4HSL. So for a visualization of our plasmid for the detect mechanism is, my apologies. This is just a visualization of our plasmid for the tech mechanism. On the right here, we have our REL-R, which is the quorum sensing receptor of Pseudomonas aeruginosa. And as it is expressed, it binds to our quorum sensing molecule C4HSL, and together they activate this pre-rel promoter on the right and express the gene GFPA1, which is a green fluorescent protein. It basically makes our microbes glow green. And here's just a visualization of our plasmid actually working. In the outside tubes here, it contains LB, which is just a medium that we use to grow our microbes and plasmids. And that naturally has some fluorescence already. So as you can see, it slightly glows a little bit of green. In the middle, we have two tubes that actually contain LB and our plasmid. And as you can see, they're visibly growing green, which means that our plasmid is in fact working. And as we made this discovery, we realized that our detect mechanism is working in fact. So we're going to go ahead and focus on our delivery mechanism, which Annie will talk to you more about.
The delivery aspect of our project's goal is to be able to have the microbe be able to actually swim towards the biofilm once it is detected. And so we're using chemotaxis, which is a microbe's natural ability to be able to swim towards a quorum sensing molecule, in this case the C4HSL molecule. And so the way that we're able to control how our microbe swims is through the production of the gene KZ. And so when KZ is not expressed, the microbe will just tumble in a circle. But then once it is expressed, it will swim in a straight line path towards the biofilm. And so then what we are doing is taking our detection plasmid, which has GFP produced in it, and instead of GFP, we are putting KZ, so that once the C4HSL is detected, our plasmid will then be able to swim towards the biofilm. And so here's an example of it where the biofilm is secreting the C4HSL, and then the microbe is tumbling until it eventually swims in straight line paths to be able to find the biofilm. Now Jonah will talk to you about how we're actually going to destroy the biofilm once it has reached.
There we go. All right, so in order to actually destroy the bacteria and the fungi in the biofilm, we found this molecule called cinnamaldehyde, and that should sound a lot like cinnamon because 90% of cinnamon is actually made of that molecule. So the really nice thing about this molecule is that it is very small, and that allows it to actually go into the bacteria's cell wall. It actually permeates through and kills off that bacteria. However, this isn't as effective against fungi because fungi has an extra little layer on top of the actual cell wall itself called chitin, and that chitin actually blocks the cinnamaldehyde from actually penetrating into the cell itself. So we actually took this thing called a chitinase, which is an enzyme that breaks down chitin, and we're going to use that to basically punch holes into the chitin and allow the cinnamaldehyde to actually permeate through and destroy the cell and the fungus. All right, so for the chitinase, we decided to go with two different kinds of chitinases. This way, it's just as a kind of a fail-safe to make sure that if one of them isn't working, the other one will pick up for that slack and still be able to punch holes in that chitin layer. All right, so the first one is chitinase B4A from Serratia mercescens, and then C1 is from Streptomyces griseus. So these are both commonly found in the actual biofuels that we have. They're both from fungi and are pretty prominent in the actual biofuel. So in order to actually produce these chitinases, we wanted them to actually stay with the bacteria itself. As Yasmin and Annie had explained, the bacteria is going to be moving towards the biofilm. So we wanted the chitinases to stay attached to the bacteria so that it'll continue to move towards the bacteria and the fungi in the biofuel and be able to punch holes in the chitin. So in order to do that, we use these things called isonucleation proteins, and they basically just act like an anchor to the bacteria. So you can see two different diagrams here, one with the B4A and one with this C1. And these basically just link the chitinase to the anchor, the isonucleation protein, and this way the bacteria will move towards the biofilm and have both chitinases attached to it. All right. So as the gene design for this, we actually got pretty lucky. One of our mentors was already working with an isonucleation protein with a different kind of enzyme called tyrosinase. So our whole goal is to take that tyrosinase and cut it out and replace it with the chitinase C1 and the B4A. And that way we'll have the isonucleation protein anchor the chitinases down to the cell. All right, so for the cinnamaldehyde, it was a little trickier, but we did find in one of the iGEM parts a team from 2013, the British Columbia team. They had already found a way of producing cinnamaldehyde in bacteria by breaking down this naturally occurring molecule called phenylalanine. And basically they created 3 different enzymes that will break down that molecule 3 different times until they get to the cinnamaldehyde molecule. However, this was 3 different parts, and we wanted to kind of make that into one single piece so it was easier to actually put into a plasmid and have it be more effective in producing the cinnamaldehyde. So we have our plasmid design here. We have the 3 different enzymes, and then we also attached an RBS, or ribosomal binding site, in front of the latter of the 2 enzymes that we are producing. This just makes them more effective and more efficient on actually breaking down and producing those enzymes to break down those molecules. All right, so we did some testing with the cinnamaldehyde because one of the main problems we have is trying to find the right amount of cinnamaldehyde to use and finding if we can actually produce enough of that cinnamaldehyde molecule to actually break down the bacteria and the fungi. So we did some testing. We did, so you can see this graph, as the concentration actually lowers, you can see the amount of absorbance of light is decreasing. So basically, we put some bacteria and fungi into some LB broth medium, the same thing that Yasmin was using. And then we added cinnamaldehyde at different concentrations and just lowered them as we went down a well plate. And then we measured the amount of light that was being able to pass through the actual liquid itself. So the more bacteria and fungi there is, the more cloudy it will look. So if the cinnamaldehyde is actually killing off the bacteria and the fungi, the actual liquid will be more clear. And that's what we were actually seeing. We were seeing this pretty big drop, right around 1 in 3,000 parts per the cinnamaldehyde versus the liquid LB and the fungi and the bacteria. So that was really nice to see that we actually got a big decrease of when the cinnamaldehyde stopped working. All right, so we did try and do some testing with cinnamaldehyde and chitinase together. And basically, we took plates, we covered them with a fungi, and then we put down a little drop of chitinase and cinnamaldehyde together, and we did them at different concentrations. However, if you can't really tell if there's any zones of clearing, there's nothing that's kind of gone. So all these dots were spots that were placed the actual cinnamaldehyde and chitinase. However, there's not really much that's disappeared. There we go. There's a tiny little bit of a circle that's going on there, but it's not much to actually conclude anything. So our results were inconclusive with that part, and we're still trying to do more testing with those. So yeah, wow. Haley will now talk about the actual medal criteria that we face for the iGEM competition to get different gold medals.
So there's a lot more to do in iGEM than just lab work. We've done lots of collaborations as well as trying to achieve all the medals. So the first medal we have is bronze medal. So we are registering and attending the iGEM Jamboree this month. The next is we have team deliverable, so it's the wiki, the poster, the presentation, which we will also have accomplished. We have an attributions page on the wiki, as well as we have participated in the interlab, which falls under characterization, and the interlab is basically just standardization of measurements across the globe. The next medal we have is silver medal criteria. So we have submitted and validated 2 parts, so 3 parts actually. We have 2 chitinase parts and 1 key Z part, as well as we've had many collaborations such as the Michigan State Meetup, which we went to Michigan State and presented and had food and talked and played games with other teams, as well as USAFA, which stands for United States Air Force Academy. They were here for 5 weeks, and we did lots of collaborations with them. And then we also have done human practices, so we've contacted local companies, like Max said at the beginning of the presentation, as well as legislators. We are meeting with Mike Turner next week to talk about legislation. and grade school presentations, which we have went to all our local grade schools and presented about DNA and did a DNA project with them explaining what DNA is. So for human practices, like I talked about, the local legislation, the biofuel companies, the National Diesel Board, which was Mr. Scott Fenwick, and visiting Beckett Marathon, and then the grade school presentations, as well as we handed out a synthetic biology form to explain what iGEM is and how to become involved. And then, so these are some of our collaborations. So this was us with the USAFA team at City Barbecue. This was the Midwestern Meetup, which is at Michigan State. And then we also did Mike the Microbe, which is basically Flat Stanley, and we sent him across the globe. So this is from a team in Israel. It's him chilling at the beach. And then we did a Skype with Singapore about modeling, which I'll get to later on. So for the gold medal criteria, we have integrated human practices, which is the cinnamaldehyde kill switch, as well as modeling our project. We collaborated with Singapore to model our KZ. So we modeled three ribosome binding sites, so it's 32, 33, and 35 for KZ, just to see which one would produce the best, because you don't want too much or too little KZ, so 33 ended up working the best. And then for acknowledgments, we would like to thank all of our mentors at AFRL for helping us and helping create our presentations in the lab work in the summer, as well as our wonderful teachers here for providing us with so much help, as well as Mr. Franz for the media and anyone else who's helped us. And that's it. Do you have any questions?